Good day, Mamel. In this video, I'll be answering the question in Cap 2, Lesson 6. Based on the discussion, what is the difference between light-dependent and light-independent reaction? In the discussion, the difference between light-dependent and light-independent reaction is that light-dependent reaction, it refers to solar energy, photons, converts into chemical energy, ATP or NADPH. We can determine light-dependent reaction in the plant cells called the chloroplast. There are also about 30 to 40% of them per cell. In photosynthesis, the light-dependent reaction takes place on the thylakoid membranes. The inside of the thylakoid membrane is called the lumen, and outside the thylakoid membrane is the stroma, where the light-independent reaction takes place. The inputs of light-dependent reaction are water and the light energy. In this process, the light reaction produces ATP, NADPH, and oxygen. There are also three parts in light-dependent reaction. These are photosystem 2, photosystem 1, and ATP synthase. Components of the light-dependent reaction is involved in two photosystems 2 and 1, an electron transport chain that are embedded in the thylakoid membrane. Light that is harvested from the PS2 causes an excited electron of the chlorophyll, a special pair, to be passed down on electron transport chain PQ, CYT, and PC to PSI. Light-dependent reaction occurs when the pigment of chlorophyll, located within the thylakoid membranes, captures energy from the sun photons to initiate the breakdown of water molecules. The goal of the light-dependent reaction of the photosynthesis is to collect the energy from the sun and break down water molecules to produce ATP and NADPH. These two energy-storing molecules are then used in the light-dependent reactions begins when sunlight hits a molecule of chlorophyll, located in photosystems 2. In chloroplast, the cycle of light-dependent reaction is where it is being made. Surrounding the chloroplast, that is a fluid, is what we call stroma. So in stroma, a pouch is called thylakoids that are suspended. The pouch of thylakoids are sometimes piled up with supported grana. Grana moves singular, and inside the membrane we can determine the particular pigment molecule called chlorophyll. And in this procedure, researchers were able to locate how light-dependent reaction is being processed by the leaves of the plants. Next, we have light-independent reaction, where chemical energy, ATP, and NADPH will be processed into 3P-glucose precursor. The Calvin cycle, light-independent reaction, biosynthetic phase-dark reaction, or photosynthetic carbon reduction cycle of photosynthesis, are the chemical reaction that converts carbon dioxide and other compounds into glucose. Light-independent reaction has three stages, which are carbon fixation, reduction, and regeneration of the starting molecule. Light-independent reaction has also stages that occur inside one of the organelles found in the plant cells, which are called chloroplast, and there are around 30 to 40 of them per cell. ATP and NADPH are used to synthesize organic molecules or anabolic pathway. The synthesis of light-independent reaction occurs in the stroma, which ATP and NADPH are being used in light-independent reaction or Calvin cycle. Light-independent reaction is anabolic, which then means that in building organic molecules from other smaller components, it requires energy. There are three steps of phases in light-independent reaction. First is carbon fixation, second reduction, and the third one is regeneration of ribulose. There are also inputs in the light-independent reaction. These are light and water, and ADP, P and NADP+ from the Calvin cycles. Inputs of the Calvin cycles are CO2 and ATP and NADP from the light reactions. So these are the differences between light-dependent reaction and light-independent reaction. How are photons absorbed and utilized in the first stage of photosynthesis? Photons are absorbed and utilized in the first stage of photosynthesis by the porphyrin ring. Porphyrin ring gives the molecule each how much volume photons can stack up solar energy in the sunlight in a particular wavelength. In the lesson, photon goes to chlorophyll ground state, next chlorophyll ground state to chlorophyll excited state. If chlorophyll can stack up a photon of a specific energy, one of the electrons will bound or hop into an excited state. This process can be called photosystem. In photosystem, the chlorophyll molecules will be placed inside of a photosystem together with other chlorophyll in the ground state. This excitation state after photons are being absorbed. This can move through around the molecule to molecule, with only one electron with an ease to back the ground state copying another photon that will eventually excite an electron in another molecule and so forth, like a pinball machine. Photons will go to the reaction center complex that contains a special pair of chlorophyll molecules. In due course, a photon will strike the reaction center complex, the high energy excited electron, rather than relaxing back down to the ground state. This will be instead shift to another molecule called the primary electron acceptor, right above the reaction center complex. After some time, H2O or water will become O2 or oxygen to become the lessened final chlorophyll. Next stage is H2O or water will be converted to O2 or oxygen to lessen the final chlorophyll. Afterwards, an enzyme will provide the missing electron to the oxidized chlorophyll across a water molecule to get it back to its normal state, which is how oxygen molecules are then produced. Summarize the process of light reaction and Calvin cycle. The process of light reaction, solar energy or photons will be converted into chemical energy or ATP. NADPH, the stroma outside the thylakoid and lumen inside the thylakoid. In light reaction, there are procedures that are a series of redox reactions, reduction and oxidation reactions, that will eventually transpire first in photosystem 2 and after in photosystem 1. When photosystems seem like they are named in reverse, it is due that they were in sequence after they were being discovered. Step 1. In the photosystem 2, the electrons are moving along in the first place with the primary electron acceptor through a sequence of compounds that are implanted in the thylakoid membrane, slightly like the electron transport chain from a cellular respiration. Following ATP, adenosine triphosphate will be an outgrowth of this procedure to a proton gradient and ATP synthase. It is also the same in the cellular respiration. It is an enzyme contained in the thylakoid membrane of chloroplast is what we call ATP synthase. This is where it generates the synthesis of ATP from ADP and inorganic phosphate, PI. Step 2. The electrons will pass through photosystem 1 and an additional electron transport chain. But as an alternative of providing ATP, photosystem 1 will outturn in the altering of nicotinamide adenine dinucleotide phosphate, or NADP+, into NADPH. So in this metamorphosis, light reaction in the thylakoid that exists in the summary by the equation, which is light plus 2H2O plus 2NADPH plus 3ADP plus 3PI, reactants to O2 plus 3ATP plus 2NADPH products, which then needs sunlight and water among the reactants and will generate some oxygen, ATP, and NADPH as products. Following ATP, adenosine triphosphate will be an outgrowth of this procedure to a proton gradient and ATP synthase. It is also the same in the cellular respiration. It is an enzyme contained in the thylakoid membrane of chloroplast is what we call ATP synthase. This is where it generates the synthesis of ATP from ADP and inorganic phosphate, PI. Step 2. The electrons will pass through photosystem 1, an additional electron transport chain. But as an alternative of providing ATP, photosystem 1 will altern in the altering of nicotinamide adenine dinucleotide phosphate, or NADP+, into NADPH. So in this metamorphosis, light reaction in the thylakoid that exists in the summary by the equation, which is light plus 2H2O plus 2NADPH plus 3ADP plus 3PI reactants to O2 plus 3ATP plus 2NADPH products, which then needs sunlight and water among the reactants and will generate some oxygen, ATP, and NADPH as products. The Calvin cycle. ATP and NADPH are used to synthesize organic molecules anabolic pathway. What Calvin cycle is using that is happening to the stroma is called ATP and NADPH. From here, it is where all combination occurs. Is similar to the citric acid cycle, Krebs cycle. It is existed as catabolic. The Calvin cycle is anabolic, which then measure as constructing of the organic molecules from the tiny ingredients that needs energy to work. Phase 1 of Calvin cycle. Rubisco is an enzyme which carbon preoccupation that happen in production. The Rubisco will eventually catches the CO2 from the atmosphere and then connects it to a 5-carbon sugar called the ribulose p-phosphate RUBP. The remaining 6 carbon molecules is considered unsteady and will break into 2 molecules of 3-phosphoglycerate. Hereafter, it then no longer have the phosphate to become glyceraldehyde 3-phosphate G3P. Part of G3P is the essential ingredient of the Calvin cycle that is proceeding to the further corridor that will give rise to the glucose and other organic compounds. And phase 3, it is the renewal of the ribulose. Few of the G3P will proceeding back to regenerate additional RUBP and begin the cycle of over again. What are all the byproducts of light-dependent and light-independent reaction? All the byproducts of light-dependent are solar energy or photons, which will be converted into chemical energy, ATP and ADPH. It also includes the photosystem 1, photosystem 2. In photosystem, there are electrons that flow starting with primary electron acceptor through a series of compounds that are embedded in the thylakoid membrane. Electrons are transported in the chain from the cellular respiration. Because of this, ATP or adenosine triphosphate will be the byproduct of this process. An ATP synthase is included because it is an enzyme localized in the thylakoid membrane of chloroplast. In photosystem 1, electrons will proceed through them. This is another transport chain. This is outturn nicotinamide adenine dinucleotide phosphate or NADP plus into NADPH. All byproducts of light-independent reaction are chemical energy. ATP and ADPH converts to G3P glucose precursor, organic molecules from smaller components that requires energy to work. Carbon fixation happen which the cause is an enzyme called Rubisco. Rubisco also captures CO2. The following 6 carbon molecules will become into 2 molecules of 3-phosphoglycerate. After that, it then drops a phosphate to become a glyceraldehyde 3-phosphate, G3P. Few of the G3P will go back to regenerate more RUBP and start the cycle over again.